The CFDE toxicology partnership is focused on elucidating the mechanisms underlying developmental defects and identifying novel teratogens. To achieve this cross-DCC team mined knowledge related to developmental toxicity from various Common Fund DCC and other resources. This knowledge includes gene birth defect associations from GWAS studies, drug birth defect associations from the literature, and gene-drug associations from drug-induced gene expression data collected by LINCS, as well as FDA-reported adverse drug interactions, drug target associations mined from literature, and chemical structure-based drug-drug similarity associations from IDG. Genetic burden scores from kits first and placental crossing probabilities for drugs were also computed. All the gathered data is stored in the reproductive toxicity, or ReproTox, knowledge graph. This knowledge graph is provided as a back-end Neo4j database and is also provided as an interactive graph-based web interface for visualizing the associations between birth defects, drugs, and genes. Nodes are identified with readable labels, as well as IDs from the Human Phenotype Ontology, NCBI gene, and PubChem. Starting with a specific birth defect gene or drug, users can explore all connections to the input node or find paths between any pairs of nodes. Edges are labeled by the resource from which the association came from, and can be filtered using a drop-down menu. Clicking on a node will display more information on the node, including the genetic burden scores for a gene, or the placental crossing probability for a drug if available. External links to resources for the entity are also provided. The ReproTox knowledge graph can also be queried for the shortest path between a pair of entities. Visual aspects of the graph, such as the layout or size, can be adjusted as well. All currently displayed nodes and associations are also displayed in table format and can be sorted, filtered, or exported. Finally, all data serializations needed to create the ReproTox graph can be downloaded from the Download Assets page and loaded locally following the instructions provided on the portal.